I'll go back and find it later, see what I can do. I'm not sure it'll be any help because the test is tomorrow. So the biological species concept really focuses on your reproductive abilities. Can a male and a female actually have a viable baby? Because sometimes you can have, we'll talk about that later when we get to what the barriers are, but some can actually reproduce together, but their species can't reproduce after them. So if you have a species that cannot continue having offspring, then you can consider one a biological species and the other a biological species. The problem with that is when you're looking through the fossil record, you couldn't tell who is able to mate with whom. So that's a really difficult thing. And then of course the ring species, we'll talk about that later, that also really is an issue with the biological species concept. And so you see here the advantage, reproductive isolation, evolutionary independence. It's kind of, yeah, so this fossils, like I said, and then the populations of ring species. It does work well for most species in most sense. The morphological species concept, they're specifically looking at traits. So you're looking at a whale has a tail. You're looking at a whale does not have legs. A hippo has legs. So you wouldn't group those together. According to their traits, they're not the same species. However, when you look at the phylogenetic evolutionary sequence of how they've evolved over time, they are in fact related to each other. So when you look at the morphological traits, you really can't see how things have evolved through time. You really can't see that things are related to each other and what are sister taxa. So it really doesn't give us, again here, it works for fossils because like in the whale, you could see the fossil, the feet, right? That's kind of how they figured it out. But when you're looking at it, you can't see it on the actual whale species. Does that make sense? I'm kind of just talking, but please stop me if something does not make sense. The phylogenetic species concept is pretty much what has been accepted because they can look at a common ancestor, and they can look at traits and how species come down the tree, and then they can look at, okay, the species has diverged. It's pretty much widely accepted as the best way to do it. The disadvantage is like it says here, there's not a lot of phylogenies because there's so many different species. It would be really difficult to try and actually get a phylogenetic tree for everything. But the terms monophyletic and, and I put this in here because he stated this, and I think it is really important. He stated it at the review, phylogenies capture the evolutionary history of a species. So the really big thing about the phylogenies is you're not just looking at a specific species the way it sits in time. You're looking at a species over the extent and course of its evolutionary history. And so that's why it's really applicable. The difference, and I'll talk about those. Let's see, what was I saying? The monophyletic, that is that's going to be in opposition to like paraphyletic and polyphyletic, so I'll talk about that. But the monophyletic is really when you get down to just having two taxa at the very end of a branch. So the difference between allopatric and sympatric speciation, again, anybody can stop me, ask questions. I'm watching the chat, so if you have something you need me to spell out a little bit better. Or if you're like, hey, I know this better than you do, feel free to throw something in. This is just what I've studied. Allopatric speciation, and I know Jillian threw this little piece in, so it's kind of interesting about the molecular clocks. So the molecular clock is just, you can estimate how long since divergence by counting the differences in DNA sequence. So you can look at the differences, how many DNA sequence differences there are, and you can kind of decide when those species diverged. Allopatry, of course, is other country. So this occurs when a species diverge from each other because they either have experienced dispersal or vicariance. Dispersal is when you have a small portion of the species, almost like the founder effect. They go to a new island or a new area. And when they go there, they start their own colony, they establish it, and then something blocks them from having gene flow back to their parent population. So now that you have this inability for genes to flow back and forth between the two populations, they'll slowly over time, they'll diverge. And as they diverge, they'll become genetically different species, and then they'll be unable to mate again. Can you guys mute yourselves, please? I'm sorry, I'm not sure who that is, but I'm getting a lot of feedback.
No way. I'm doing homework right now.
How do I work the remote?
I'm doing homework.
I'm not sure who that is. Let's see, how do I mute people on here? Well, it seems it's taken care of for me. Vicariance happens when it's not necessarily that a population founds a new colony, but something happens that splits them. Say a volcanic eruption, a continental split. Something happens that divides the species into two, and it has to be something that prevents them again from having gene flow. That's really the key phrase when you're looking at allopatric speciation, is that there is an inability for there to be gene flow because somehow the parent population has been separated from its offspring, and then they become distinct from each other. Pangea, yeah, that's a good example. That's when the continent split, and then yeah, you would definitely have different species popping up there. And there's a lot of little examples here. I'm not sure he'll ask for any examples, but they're there. In contrast to allopatric, you have sympatric speciation. Sympatric speciation, it occurs when there's really, it doesn't seem like there should be a reason why there's this inability to interbreed, but like I think everybody did the anoles experiment. The anoles began to fill different niches of the environment, and when they filled niches, then selection acts on the different populations, and then they're unable to mate with each other. So one of my children did not go to bed, so we'll have a little bit of interruption once in a while. Anyway, so the sympatric speciation, just Jillian marked out here, disruptive selection, niche differentiation. Those are really important because something disrupts them, they fill different niches. And I think you can see a lot of that when he's talking about the mass extinction, and then suddenly you have this explosion of, well, we'll talk about that later. I'll get into that, but that is definitely, they're still in the same region with sympatric, they just no longer interbreed. This another way that it happened is this polyploidization. Polyploidization is when you have, say, non-disjunction occurs, so you don't have the chromosomes splitting correctly, so that you get too many chromosomes in one of the offspring, and then that species continues to have that number of chromosomes in its offspring, and it's unable to then go back and mate with the parent population or breed with the parent population. So that's just has to do with the chromosomes it has more chromosomes, and so that difference in chromosomes just makes it incapable of interbreeding with parent populations. Am I going too fast? Somebody slow me if you need me to. I think this is a really good part too when he's talking about the no geographic isolation. It's a good thing to know because the sympatric individuals, they live in the same geographic area. They have not moved. They're still in the same area in a sense. Divergence, something happens to them to cause them to diverge. Selection happens on them, and then they're isolated from each other. So that's a good thing. That's a good one to know. And then here's a bunch of examples for you. Meiosis, this is that polyploidization again, basically. There's errors, so you have the non-disjunction or mitosis combined. So the self-fertilization, or so that's when they have all the extra chromosomes, and then they're then unable to reproduce with parent generations. That's really important about this, is when you look at, are they able to reproduce with the parent's generation? He talked about reproductive barriers. There's two real big categories, which is the prezygotic barriers and the postzygotic. Prezygotic just means before you have a zygote, so before the sperm and the egg make a zygote, There's barriers that prevent the sperm and egg from coming together and making an offspring. Post-zygotic means somehow the egg and sperm do come together, but the offspring is either inviable or sterile. These are good to know. I'm pretty sure he's going to ask about one of these. So you have the temporal, just basically means they're isolated because of time when they breed. So they breed at different times of the year, different times of the day, just different seasons. They're incapable of breeding because they breed in different habitats. They need different types of environments to breed. They have behavioral differences, such as their courtship displays, how they mate, or how they try to get the female. Mechanical, this is just basically happens when your reproductive organs do not fit together. So they actually cannot fit in structurally. And then gametic barriers, the egg and the sperm, they cannot fuse together. So maybe sex occurred, but the sperm and egg cannot fuse to make a zygote. And then post-zygotic, those are pretty simple. The hybrid inviability, so the offspring is inviable. So actually, when the hybrid invariability occurs, there is no actual offspring produced. Like the egg and the sperm and the egg come together, they make a zygote, but in development, before that offspring is even born, it dies. The hybrid sterility has to do like with the mules. They're sterile, they can't produce. So that stops that generation. These are the different things. So say you have two populations that have been separated, but they come back together somehow. You have this list of possibilities that could happen. First of all, you could have a fusion of the populations. So maybe they come back into contact and they just come back together and they interbreed and they're fine. There's no difference. So that's a possibility. Another possibility is a reinforcement of divergence. In other words, they come back together and it's like two walls hitting, they cannot interbreed, nothing can happen. So it just reinforces the fact that they are now two divergent populations. You have a hybrid zone, which is where they come together and maybe these species, they start to overlap where they do come together, but you still have kind of two distinct species, and then in the middle you'll have kind of a new species forming, but they're able to breed across the line, interbreed. The next is extinction of a population, so you have them coming back together, and one is just more fit than the other and wipes the other population out. The last is the formation of a new species. This is in some ways a bit like the hybrid zone because they come back together and they start to interbreed. But then what happens is the offspring is different and cannot reproduce with the parent generation. So then you have three populations now. So the ring species, this one's pretty straightforward. You have, say you have population 1 and it can breed with population 2 and that can breed with population 3, they can breed with population 4 and around the circle, and then all of a sudden you get to like 7 and 7 can't go back and breed with 1. So it kind of, it has an issue, they have an issue with this, with the biological species concept, because you have these species that can interbreed all the way around, but then these populations, I shouldn't say species, I should say populations, these two populations can't interbreed. So are they different species, or at what point did they become a different species that they no longer can interbreed? So your biological species concept just is blown out of the water with those. Phylogeny. So the phylogeny is the history of the evolution of species or groups. That's just your basic, you've got your root, comes down to your nodes, your branches, and that comes out and builds your phylogenetic tree, and at the end are your taxa. I think that's all pretty just straightforward. That's pretty much just definitions on a lot of this. And the really important thing to know, and I want to point this out because I think it's probably going to be a question, phylogenetic trees are just hypotheses. They're not proven. They're just a set of data that we have taken, and we've built this phylogenetic tree to the best of our abilities, and we're going to take that and we're going to test it against other trees and other concepts to try and find the one that fits the best. But at no point is it a proven fact. This is not proven. And need to know that the phylogenies, they compare the ancestral traits to the derived traits. Ancestral traits are what you're going to see in your ancestors coming down the line, your common ancestor. And that has to do with the homology. And your derived traits are what you're going to see in your taxa at the other end of the phylogenetic tree. So that's actually what you're going to see in the species that are current, or that you could have a phylogenetic tree that ends with a species that has gone extinct too, but it's just the farthest on the right of the phylogenetic tree. And this is important to you in here, made with the one data set. So you're just looking at morphological traits, different traits, and those could be similarities in observable traits or similarities in DNA traits, or there could be different things that they base the phylogenetic tree on, but then those are tested against other morphological similarities. Because I think the good one, we'll talk about it again, is the birds and the bats. Is okay, well, we're going to put these two species together as sister taxa because they both have wings. But now we're going to take this phylogenetic tree that has them as sister taxa, and we're going to compare it to this other morphological tree that compares the bones in the wings. Well, now that we've compared this phylogenetic tree with them being sister taxa to this phylogenetic tree, we can see that they're not sister taxa because the bones in the wings are completely different. And there's other things like they produce milk. Okay, now they're not the same species. Whereas on this phylogenetic tree, we thought they were based on their wings. I hope that makes sense. Y'all are very quiet, so I'm okay with that. I hope that y'all are okay with what I'm saying. The convergent evolution, and I threw the definition of homoplasia in here because it's a good thing to know. Homoplasia is a character shared by a set of species, but not present in the common ancestor. So that is when you have convergent evolution. You've got bats there. The bats are evolving wings over here from this ancestor, and birds are evolving wings over here from this ancestor. They're evolving convergently, but they don't share a direct common ancestor. I'm sure somewhere along the phylogenetic tree they would, but they don't share a direct common ancestor. So they're evolving convergently. If we were to use that phylogenetic tree where we have birds and bats as sister taxa, we would have a polyphyletic grouping. That's because we grouped two things together that don't belong together. They don't share a common ancestor. So that's an error. It's an incorrect. Polyphyletic trees or polyphyletic groupings are incorrect. In contrast to that, you could have a paraphyletic group, and a paraphyletic group is when you eliminate an animal because it doesn't have a trait. So you eliminate it from your phylogenetic tree. You say, okay, well, whales, they don't have legs, so they're an outgroup of these other mammals. Well, you've created a paraphyletic grouping because you've left out an animal that actually does in fact belong in that phylogenetic tree. They belong right next to hippos because they look like they're, well, from what they can see so far on phylogenetic trees, they seem to be sister taxa. So polyphyletic, you have things that are grouped together that don't belong together. Paraphyletic, you have things that are not grouped together that do belong together. And that often happens because of the paraphyletic often happens because of an evolutionary loss of a trait. And evolutionary loss of a trait just means that that species evolved in such a way that it no longer needed that trait, or it lost it somehow through some kind of selection. It lost that trait, and that's an evolutionary loss. I'm not going to go over these because I think these are pretty self-explanatory. They're just, unless somebody, tell me if you need, but these are the definitions of what's going on here. The only thing I'll tell you is about polytomy. Polytomy is important because that's what we saw in the fungus. It's when you have too many taxa coming off one node point. That happens because we don't have enough data to really resolve what's going on there. We don't know who exactly is related to whom. So they're just kind of all branching together from the same node. Polyphyletic group. That's the polyphyletic we talked about. So there's some definitions, the polyphyletic, paraphyletic, and monophyletic, if you need to come back and look at those. then you have, okay, here's the definition. The monophyletic is what we're looking, when we're doing a phylogenetic tree, we are looking for a monophyletic group. That tells us the closest that we can get to who in fact are sister taxa and who are the little branching family that they belong with. Synapomorphies are just the shared characteristics. So hair in dogs and humans, that would be a synapomorphy. Homoplasia, we talked about that. It's the character between two, it arose not from a common ancestor, but from two different ancestors. They evolved convergently. Oh, I didn't go back and I'm sorry, Jillian, I didn't define evolutionary loss there for you. Again, these are just basic definitions. Sister taxa are the two. You really want to see just the two coming off that final branch of the phylogenetic tree. But if you see more, they're still sister taxa, common ancestor. And this is important too. I think he kind of stressed this, so I think he might ask about it. We cannot infer time or amount of divergence unless it says it explicitly in the figure caption. So when we're looking at phylogenetic tree, we can't think, okay, this line is this length because it took 500 million years for these species to diverge. We can't do that. It's just, a phylogenetic tree is arbitrary. It's just looking at the taxa unless they specifically say this length means this amount of time or this number of nucleotide differences in the DNA sequence. Okay, these are basic, these are really easy. The photosynthesis occurred, you need to know that they occurred in this eon, the Archean eon. And that they're able to prove that that happened because of the spike in O2. It changed the rocks. Like, you didn't have iron in rocks until you had oxygen in the air. So that's what they were able to tell. And then the multicellularity, he just wanted us to see, okay, this happened in this era, and then our eon, and then plants and fungi, and then animals coming to land occurred in the Ordovician eon. Hope I said that right. And then he talked a lot about the extinction. This one is important when you think about the species filling niches. I'm going to see if I can pull up real fast. I have, see if I can pull up the PowerPoint that shows them filling different niches here real fast. Jeez. I think this is a good one to look at. Okay, here's it. I'm gonna share this one real quick. Okay, so just show the adaptive radiation. So in the background, we always have extinctions, and you can see it on this, I think the bottom one, I can't really read it that well. I guess I should open it up and look at it a little bit bigger. Okay, the top one is our extinctions, and the bottom one is our adaptive radiations. I hope can you guys see this screen, the PowerPoint that I'm looking at? I'll take that as a yes. So what you have is you have all these little tiny extinctions. They happen constantly. Animals are constantly, species are coming about, they're going extinct, they're coming about, they're going extinct. It's constant throughout time. But you have these points where you have these mass extinctions that occur. And you can look at the adaptive radiation of species. So when you say you have like a massive predator or another species that dies off, suddenly you have other species coming and they're able to fill these niches that were occupied by other species. So you'll see an extinction happen, and that's a spike is an extinction. And then right following it, you can see that there's an adaptive radiation occurrence happening. And that's like, we can see the really tall spike on the graph is that new adaptive radiation right after a mass extinction. And those species are just explode because they're just able to fill all these niches that have been wiped out when this mass extinction occurred. Okay, I'll go back to this guy. Okay. I think it's interesting that he said that extinctions are the fate of all species. Maybe so. Maybe there'll be nothing left at the end. And they usually, so your big extinctions usually occur due to some sort of climate change. So you have the volcanic eruptions or a meteorite hitting the earth, or all those kind of different things, those really big occurrences. It has to be such a huge occurrence that it, like with a volcano, the ash spews up and it covers the sun and it cools the planet, and suddenly you have animals dying off because of that. Or, so there's different, those kind of things. It has to be so dramatic that it changes the climate of the environment and the animals cannot live in that environment. So your biggest extinction was the Permian extinction. That was the one we were looking at. That was followed by that really massive adaptive radiation spike. The KT extinction is when the dinosaurs died off. So that's the most well known. And our recent one is what we're in now. We're in the 6th mass extinction of our planet, actually, as far as we know. All of this is subjective to the information that we have at the current point in time. The adaptive radiation is when you're surviving lineages, then they radiate out into film new forms, and they fill new niches. So there'll be a lot of species popping up too. Chapter 26, bacteria and archaea. So the catch-22 is really simple. Basically, if you can't grow it, it doesn't exist. That was what they thought, because they only had one specific agar that was like kind of created to grow things in our body. They were trying to grow it on the agar. Well, if it doesn't grow there, then they thought, well, it didn't exist to us. If we couldn't grow it, we didn't know it. And so when we got the technology to then see these microbes, then it kind of blew our minds, because we could see there were so many more bacterial populations than we ever imagined, because we couldn't grow them all. Koch's postulate's pretty straightforward. You have to have evidence to establish that one microorganism does in fact cause a disease. So you have to isolate the microorganism. When you see a disease pop up in an individual, you have to take out the organism. You have to isolate it and grow it in a pure culture, which was that agar culture. Then you use that culture and you give it to another species or another animal. And that disease has to then grow in that animal. Then you take that organism back out of the disease animal, and then you have your, not your proof, because again, science is not proof, but it supports the evidence that that microorganism does in fact cause that disease. And when you look back at the history of germ theory, this was kind of like the nail in the coffin for the idea of germ theory. It really led us into understanding that in fact microorganisms do cause disease. Okay. This one's kind of hard to wrap your head around sometimes when you're talking about the bacterial cell wall. So just pause me if you need more on this. Every bacteria, they have a cell wall that's made up of peptidoglycan. Peptidoglycan is melted. It's eaten away by antibiotics. And antibiotics, to relate it, because all of biology is kind of all tied together, antibiotics are created by fungus. The fungus creates the antibiotics to kill the bacteria so they can compete for the same nutrient sources. So the antibiotic, yeah, it's kind of a neat little thing to realize that fungus and bacteria, they grow in the same environments. Bacteria grows a lot faster than fungus. So if bacteria were allowed to just grow, they would just grow so astronomically that they would eat up all the resources that the fungus needs. So the fungus produces antibiotics to then have the ability to survive, because they grow a lot slower. So they survive in those niches. That bacteria, so some bacteria just have I'm going to pull up this slide too, so we can kind of, let me see if I can find it really quick here. I don't want to take up too much of your time. If you guys have to go, just let me know. I'm or just jump off. It's totally fine. I'm going to try to get the bacterial cell wall. Look at first. There's 8, 9. And of course, my computer is a little slow tonight, so the internet is slow. Okay, sorry about that. I'm back. So the bacterial cell wall, we can kind of see that this is the peptidoglycan is I don't know if you guys, can y'all see my cursor over the screen?
Yes.
Yes.
Okay, awesome. So this is the peptidoglycan in here, and this is what those antibiotics melt through. They have to melt through that peptidoglycan to get into the bacteria to kill it. Some of the bacteria actually have this outer membrane, and this is not made up of peptidoglycan, so it protects the bacteria to a great extent from the antibiotics. They can't really just melt through that. And this is where you get your gram positive versus your gram negative, because when you do this peptidoglycan is easily stained, and so it turns that purple color, whereas the outer membrane is not easily stained, and so it turns pink instead. That's really important. And while we're here, I'll just show you. This is the cell wall of this bacteria. The endospore is really important because that when the environment becomes inhospitable and the bacteria can't survive and thrive in that environment, it'll kind of wrap its DNA, its chromosomes, everything inside of the endospore, and then it'll lay dormant, and the bacterium itself it'll just dry up and desiccate. But your bacteria is still alive basically inside this endospore. And then when it's good favorable conditions, that bacteria can come back to life. It's like botulism endospores in honey. They could just come back to life, and it's kind of scary because you never know that they're there because they're just they hide so well inside those endospores. Back to the study guide here. Okay. Covered that. Okay, so this is where it's a little confusing, so I'm going to try and talk it slow. Because bacteria plays a really big role in the nitrogen cycle, and nitrogen is necessary for plants to grow. Plants cannot grow without this. So for many, many years, until we had the technology to eliminate this, it was a limiting factor. We could only grow as many plants as we could based on how much nitrogen the bacteria could bring into the soil. And this process is called fixation, of bringing that nitrogen down into the soil and making it into a compound that the plants can then use to grow with. And like I said, plants really depend on that. When we use the Haber-Bosch, this new process that allowed us to, through fertilizers, to circumvent this whole bacterial process and just put nitrogen straight into the soil for the plants, that excess nitrogen is then washed into rivers and streams, and those rivers and streams wash it straight down to the ocean. And when it hits the ocean, I'm going to actually pull up this slide too, because this is a good slide to know. I'm going to see if I can find all these real fast for you guys. We need to come back to that. That's important. Okay, here's the nitrogen cycle. And this is kind of how it works. And so you can kind of see the way things go. And this is when it goes into the rivers and streams. Oh, I bet I think he talked about it on the other slide, didn't he? Yeah, he talked about it on the other slide. Because this is how I'm skipping all this. I'll come back to all this. Don't worry. I just want to show you there's this connection that's really important to understand, and it's right here. So the primary producers are these unicellular eukaryotes. They make up the very base of the food web. And so when he's talking about the importance of bacteria in this whole nitrogen cycle, what ends up happening is those fertilizers, they run down into the ocean and they feed these primary producers. And so these primary producers, they get all fat and happy off of all the fertilizers, and they have these huge explosions, and sometimes they're red, and that's when you have your the red tide. It's huge algal blooms. And so those primary producers, they're eating up that nitrogen and then they die. And when they die, because that's just their life cycle, there's no cause for it, just their life cycle, they die and they start falling down to the bottom here, where these scavengers begin to decompose them. Well, these scavengers, these guys produce oxygen, right? They use carbon dioxide and they produce oxygen because that's their plants. But when they come to the bottom, they're eaten and decomposed by these scavengers who use oxygen and create carbon dioxide. So when you get this huge amount of primary producers that are decomposing down here and these scavengers are eating it up, they're using up all the oxygen and they're multiplying and they're using up all the oxygen down there. So suddenly you have a dead zone because that portion of the ocean is completely devoid of oxygen. Well, not completely, of course, but it's devoid of oxygen enough that other things can't grow and live in that environment. And so it's called a dead zone. And so that's why it's important to understand that when the bacteria did the fixation of the nitrogen, and the bacteria were what gave nitrogen to the plants, we weren't having these dead zones and these red tides because there wasn't the excess nitrogen flowing down to the ocean. I hope that makes sense. So it's kind of all connected there when he talks about the point of all that. Okay, the DNA transfer, let's go ahead and look at that slide because that one is an important slide too. Talk about all this stuff is really important. These are definitely going to be, oh, it was back in that bacteria one, wasn't it? Forgive my slowness. My day has been very long. It usually is with children. Okay, so these are important slides to be familiar with too. These are how you have transfer of DNA. And so this one's really important with so you have the cell that it dies and it releases its DNA into the environment. Another cell comes, or another organism comes, and it picks up that DNA from the donor. So this guy is the donor. This guy is the new host for it. He's the host cell. He takes that DNA, he incorporates it into his DNA, and suddenly you have this new cell, totally new cell, and this is called DNA transfer by transformation. It's taking in DNA and transforming it into something new. Transduction, DNA transfer by transduction, you have these bacteriophages. They kill bacteria. They're like viruses that kill bacteria. So they go and they kill the bacteria and they pick up this DNA from this bacteria and they take it over to this other bacteria or the other cell, and they inject the virus with that new DNA into the cell. That cell picks it up and now it's transduced. It has this new recombinant DNA. The conjugation, it can involve either plasmids or chromosomes. Plasmids are your ring structures. Chromosomes are your little squiggly spaghetti structures. So they can either take, and they can just do lateral transfer. They just basically share their plasmids and they copy it, and suddenly they're like clones of each other. And they can do that with both their plasmids or their chromosomes. Simple there. Okay. So, and then this just lists it out. I just typed out the list of how that happens on each of those. So that's pretty easy. You can just kind of look through the list. Biofilm, I went through the little list and I just kind of wrote down what ends up happening, because you have these like little tiny free living prokaryotes. They bind to a surface and they become attached, so attached to that surface. And then they begin to grow and divide, and there's this new matrix of all these little tiny prokaryotes. And this is now a single species biofilm. It's just got one species. It's a biofilm, but it has one species of all these little prokaryotes that are stuck to a surface. But then that biofilm that's just one species can send out little signals and attract other organisms to it. And then other organisms can come and they become trapped in the matrix. And this creates a mature biofilm because it has a couple of different species that are all connected together. Oh, I didn't define archaea here. I'm not really sure what the definition of archaea is, but it is more closely related to us than to the bacteria. Before they thought that it was closely related to bacteria because it's a single-celled organism, and so is bacteria. It also, I believe it has a membrane-bound nucleus. Let's see. I should have it on here. Okay, so no, it does not. So if you look here, the difference is we, or eukaryotes, have this nuclear envelope. They do mitosis, they do meiosis, they have mitochondrial cellular respiration, and they have a cytoskeleton. Here, that's when all this happens. Archaea doesn't have any of that, any of those synapomorphies. So they don't have any of that. They don't have the nuclear envelope or anything. So that's it. That's a good thing to know. So they differ from us, but they're still our sister taxa to an extent. And then, of course, they say that eukaryotes arose due to the envelopment of a bacteria by archaea. These are the three, there's three major events. So we'll go and look at those major events that occurred because they're all very important. So first, you have this you have this bacterium, and it gets swallowed by a host cell. And so now you'll see there's two distinct cell walls, because they are distinct cells. But then they develop this endosymbiotic relationship where one is making ATP, the other one's giving it pyruvate oxygen, and it's just this cycle. They help each other out. And this is the first one. We have this mitochondria, and the mitochondria is it's cellular respiration. It does ATP. That's why mitochondria, they can reproduce independently of the rest of the cell. They have this extra cell wall. So this all is really important to know, to see. This was the very first of the three major events that occurred, and so now you have cellular respiration. The second major event that occurred gave us the chloroplasts, and that's when you have a cyanobacteria. So you can see those different cell walls. It gets taken in by this eukaryote, gets swallowed up in there, and now now it's another symbiotic relationship where they're taking care of each other. So that's there's your second major event that happened. So remember, and he kept stressing this, the mitochondria is in all eukaryotes. So plants are eukaryotes, so they have mitochondria as well. They did the same thing with that, where they took in that bacterium and they started doing cellular respiration. But then plants, unlike us, took in the cyanobacteria, and it became the chloroplasts in the plants. Oh, I'm sorry, I missed somebody's question. I apologize. Yeah, mitochondria was a bacteria. Let's see, go back up there. Yeah, it's this little bacterium here. He went in. So you have a bacteria, you have a cyanobacteria for the chloroplasts. And then the last really big event, so the third event was the enfolding around the nucleus. So at first, see, archaea, they don't have, and this is when we diverged from archaea, because archaea doesn't have a membrane-bound nucleus, but we do. And so we had this enfolding in on our nucleus, and then it created a nuclear envelope. And after that, we had the ER and the Golgi apparatus and other things began to develop. Okay, I'll go back and look at the, oh, and this is another thing. So there was the primary endosymbiosis of that, because they found these plants, like these are the blue ones, the secondary that do secondary endosymbiosis are all your blue lines. They found that their chloroplasts were surrounded by like 4 membranes. So they didn't understand why there was these 4 membranes, and they figured out that what happened was this cell ate this cyanobacteria, then this cell then took in this entire cell, and suddenly you have one inside the other. And so that's why you have this 4. So those are your secondary endosymbiotic relationships, and those are like your algaes. And so look at the blue. The green did it the primary endosymbiosis. So they, all of these though, the blue can all do photosynthesis, no matter if they're primary or secondary, they can all do photosynthesis. And this is going to list out those 3 major events, just so you know what they all are. And then when asked what lineages can photosynthesize, he said, don't worry about the multicellular lineages here. He said he didn't talk about those, but the lineages that can do photosynthesis are the ones that underwent primary and secondary endosymbiosis. They can both do it. Oh, and here's where I talked about, I already talked about this, how the unicellular eukaryotes play a major role in the food web. That's when you had those primary producers consuming the nitrogen that was coming off the farms and then creating those dead zones. So they're the primary producers. They do a lot of work. They're really important. They make up the base of the food web. They're really important. But when you have too high levels of them, that's when they become a problem. Oh, these are better seen on here. I think. Well, you guys are hanging tight, so I guess this is not too bad. Let's see. Oh, and that's your dead zone. So if you see that one, that's the dead zone. See, okay. So here is the 3 modes of the sex history. You really need to know diploid dominant, haploid dominant, alteration of generations. Your diploid dominant, you can see it through the red is showing when the cell here is diploid. It's only haploid for this when it undergoes meiosis. So like in us, our cells undergo meiosis right when they're about to, so then we make our egg and our sperm, then fertilization happens and it's a zygote again. It's a diploid again. So they're only haploid for the time that they're a sperm and an egg, and then they're diploid again. So our cell cycles are diploid dominant, and diatoms, animals, it's not just us, it's other creatures too, but we spend most of our cells spend most of their life as diploid. Haploid dominant, you can see it's the reverse. So we have a lot of blue. They spend the majority of their life as a haploid cell. So, and if anybody does not know what haploid and diploid means, let me know. And so the haploid over here is the N, diploid is 2N. They spend all these, so these are, what are these creatures? Dinoflagella? Yeah, the dinoflagellates, fungi. They are mostly hanging out with just this one set of chromosomes. They don't have all of them. And there's, it shows it here. It can either happen, cell cycle can expand through sexual reproduction or asexual reproduction. That's not a limiting factor on either of these. These can also do sexual reproduction or asexual reproduction. You just need to decide, is it mostly diploid or mostly haploid? Which one is the dominant? Where it gets interesting is with the alternation of generations. This can happen in 2 instances. And so there's this instance and then there's this instance. But here we have the sporophyte. This is 2N. So here we've got this, they're diploid here. They undergo mitosis. We have a sporophyte. Here's our structure that releases these little, somebody help me, spores, sorry. They release the spores, then they undergo mitosis, which is different than meiosis, and they become these male and female gametophytes. The interesting part is that in this specific cycle, the gametophytes can look exactly like the sporophytes. Sorry, did you have a question? Okay. So they can look exactly alike. And then you have, so here's your male and here's your female. They have their gametes, they come together, the gametes, the sperm and the egg come together, they undergo fertilization, and then they make the zygote, which is diploid, and they undergo meiosis again. So it can go, it's interesting because we'll learn about plants more, he said later on. But so one generation, this entire generation of the plant is diploid. Then this generation of the plant is haploid. The next generation is diploid. The next generation is haploid. So it alternates. The parent is diploid. The child is haploid. The next child is diploid, then haploid. And so it's a different, each generation is either haploid or diploid. In this set, the alternation of the multicellular haploid and diploid forms, all that is a sporophyte looks different than the gametophyte. So you have the sporophyte here, making its little spores. It sends it out. They undergo mitosis. You have your male and female again, but they don't look anything like the parent generation. But we're still going through the same alteration of generations. So we have one generation that's a diploid dominant, one generation's haploid dominant, next generation's diploid. And so it just alternates between the two. Fungi. Fungi get their energy. This is an interesting one with the fungi. I like fungus. It's interesting. Some things that are interesting to note. He was talking about how this is your paraphyletic group. Polyphyletic groups. You can see there's just way too many branches coming off. So they need more data to resolve that. That's a paraphyletic group. It's polyphyletic. I'm sorry, polyphyletic group. He wanted you to know the definition of yeast and hyphae and mycelia. It's right there. It's also in the sheet too. What was I looking for? I was looking for this. Okay, so fungi get their nutrients because they secrete, one thing that they do, they secrete enzymes outside their body. Those enzymes then digest whatever they're wanting to eat, and then they absorb the nutrients. So they can either be this saprophytic, where they eat just the dead stuff. So they're on trees, they send the enzyme out into the wood, they digest the wood, they absorb the soluble nutrients. That's one way. Parasitic is another way that they get their nutrients, and parasitic is when they live on the host, either a plant, animal, whatever. They live on the host and they feed off the nutrition of the host. They don't get anything the host doesn't get anything in return. In fact, they're usually hurt by the fungus. Not in all cases, but a parasite, they get their energy from the host, but they give nothing back to the host. Predatory, he said in the predatory, let's see if I can go down to this. So here's your parasitic. You can see that when they're just stealing energy, nutrients and energy from the plant. Here, this is just really simple. It's not really something to know much about, but you have the predators. They actually have these little structures where they can catch their prey, and then they eat away. They like these crazy ants. They use them as hosts, and they just eat away at them. The last way that's really important is through mutualism. Mutualism, this is the mycorrhizal fungi form, and that's all the white roots that you see. They, in this mutualistic relationship, the fungus help whatever is their host. So in this case, when that fungus is reaching out from the plant roots, it's grabbing in more nutrients, and the plant is able to absorb more nutrients through all of that, the network, and the plant is actually helped. The fungus gets their nutrients from the plant doing its like photosynthesis and everything, so they help each other, and that's the biggest thing about the mutualism. Then he said just to find the yeast, the hyphae, the mycelium, fruiting body, the mycorrhizal. That's what I talked about, the mycorrhizal being that web-like stuff that comes off of plants. So those are just basic descriptions. And of course, we talked about how they're parasitic. And this kind of goes into, I wrote out those slides, so you had the germination on the leaves. And so you want to read those. That's how they're predatory, and that's how they're mutualistic. Lichen is just the symbiotic. It's just it's that stuff that you see on the trees is just a symbiosis between the fungi and algae, and sometimes you have bacteria on the top of it, but it is two species living together. Why is fungal sex weird from the perspective of animals? This is really, I just wrote it all out. They can undergo, they don't have sex, so there's no male or female. So anybody can reproduce with anybody, and they're opportunistic. So they can either reproduce sexually with another organism or with another fungi, or they can reproduce asexually. So anyone can mate with anyone. Cells, they fuse, which is the plasmogamy. That's when cells just fuse together. But karyogamy is when the actual nucleus of the cells don't fuse. So then you have a cell that's fused, so they share cytoplasm, but you have two nuclei inside of the cell. And those nuclei might one day fuse, but it could take forever. It could take years, or it may never happen. They're haploid dominant. Diploid conditions exist of one or few cells. So you might have for a moment in time, they'll become diploid, but then they'll go back to their haploid. They're definitely haploid dominant. And the heterokaryotic is when just means that the cells have more than one nucleus. That's just the definition of that. Oh, I thought there was one more question. I must have skipped the one where it said how are fungi like us. I must have skipped it here. Oh, I see. I missed it here. How are they like us? They're like us because they're closely related to us on the phylogenetic tree, but they're heterotrophs, so they have to get their energy from outside themselves. Whereas plants are autotrophs, they make their own food. Fungi have to go and they have to get their own food. And they're also like us because they digest the nutrients, but they're different because they have to do digestion outside their body, whereas we do digestion inside our bodies. Well, that's everything, I think. Does anybody have any questions? Any of that not make sense?
No.
Okay. Does anybody want to go over anything anymore, or is everyone? If everyone's good, then I know somebody asked me to look over the phylogenetic tree thing, so.
Yeah, could we, at least for me,
Okay.
the tree.
Okay. If there's a way to share this, let me see. While I was chatting here.
Were you recording this? Because like I came in later, so I didn't get to catch what you were doing in the beginning, or like the review you were going over in the beginning.
Yeah, I recorded this on my computer. Now I have to figure out where the recording went to because I just hit record. So, I hope this. Yeah, I hope it helped you guys. I'll look where it is and try and share that recording tonight.
Thank you.
When we're done here. Sure thing. And I will go over. Yeah, I really appreciate being. No, bio is actually not my favorite subject. I do like it. I enjoy it a lot. I'm not really into the evolutionary stuff. It's kind of weird to me. It's just weird to think about. But see, recorded lecture should go to your email. Okay, thank you. Awesome. I'll share the link. And I will go over, if anybody wants help going over the phylogenetic tree real fast, I can kind of just talk about that. Yeah, don't forget to do the extra credit. That's due tomorrow. It was pretty simple, not too hard. Okay, so the phylogenetic tree, I wish I could somehow show you my work, but do you have the worksheet in front of you? You can kind of look it over. If not, you'll probably want to print it so you can kind of see what we're doing. Hello?
Okay, yeah, I got it.
Okay, that's cool. Okay, so the first thing that you're gonna want to do is lay out your taxa and your synapomorphies, and just lay them out in the order. So your taxa are the easy ones. Figure out the synapomorphies is a little bit more tricky because you have to go through and read them all. But on the top, you should have all your taxa. Running down the side, you should have your synapomorphies, which are the chlorophyll A and B, true leaves, seeds, stomata, vascular tissue, flowers, parallel veins, and reticulated veins. So once you have all of that listed out, then you're going to go through on each of your columns and you're going to see if your taxa has that specific trait. So the first one is green algae. And when we see green algae only has one trait, we automatically know that's our outgroup because it's not going to share any other traits with any other of the species on our list. So that's our outgroup just for comparison. And then when you go through your list, I'll just lay them out there for you so that you don't have to do it all and then you can kind of see. Pine trees have, wherever you have a trait, put a 1. If you don't have the trait, put a 0, and then you'll kind of tally them at the end. So for pines have, you'll put a 1 for chlorophyll A and B, for true leaves. I'm sorry, do you have this all written out? I am just charging through this if you need more time to write out your synapomorphies.
Oh yeah, I got it.
Okay. So they have true leaves, they have seeds, stomata, and vascular tissue. The last 3 should be 0s. For club moss, you should have 1 under chlorophyll A and B, stomata, and vascular tissue. Everything else should be 0s. For grass, you should have a 1 under chlorophyll A and B, true leaves, seeds, stomata, vascular tissue, flowers, parallel veins, and then you would have a 0 at reticulated veins. Good night. Okay, let's see, grass, when we did that one, rose should have a 1 at chlorophyll A and B, true leaves, seeds, stomata, vascular tissue, flowers, and reticulated veins. Moss should have a 1 at chlorophyll A and B, and a 1 at stomata. Lily should have a 1 at all of them except for the reticulated veins. And ferns should have a 1 at chlorophyll A and B, true leaves, stomata, and vascular tissue. All the rest are 0s. And then sunflowers should have a 1 at everything except for parallel veins. Now, when you go to the bottom, when you look at the way that I think it's easiest to look at this is then tally your number of 1s, and you're going to kind of see how your grouping is going to go. So algae is going to be your outgroup. And at one end, I wonder if I annotate. Oh, I have an annotate. I'm going to see if I can draw this for you real fast. I'm going to find a blank page here. Here we go. Okay, I'm going to annotate this. So you have your, this is your root, right? And then you come up here and you have all the way, you want to go all the way to the end, you have your green algae. And that's our outgroup. This is really hard to do, so I hope this works. And then you're going to come here, and this one's going to drop down, but it's going to have 2 that come off of it. Let's go all the way to the end. And this is going to be your next one that only has 2 of the synapomorphies, which is our moss. Okay. And then we're going to have another one that comes down. All right. And then it's going to come and it's going to have 2. Oh, that was ugly. So it's going to be the one that has 3, which is your club moss. Okay. And we're going to have another one here, and it's going to come down. This is the ugliest phylogenetic tree ever, but I hope it helps. And then you're going to have your grass. Oh, not your grass. I'm sorry. That has 7. I didn't mean that. Your next one that has 4, which is your ferns. Okay, so then we want the one that has, so this one had 1 synapomorphy, right? Moss had, I'm just checking myself here, moss had 2, club moss had 3, our fern had 4. And then we're gonna come and we're gonna do another one. We're gonna come down, and this one comes in. And then we have the one with 5, which is the pine. Okay, and then it's gonna come this way again, down, and then here's where we have, so then you notice that you have 4 that have 7. So what we have to do, I'm gonna go ahead and stop annotating for a second and pull this down. So then what we have to do is decide who of these two are, oh, darn it. It's gonna be right in my way here. Which of these two are sister taxa and which are not. So when we're gonna do this, we know that at the end here, there's gonna be a branch like this. And then coming off of here, there's gonna be another branch like this. Okay, so now we have to decide what goes where. So when we look at the traits, we can see that grass and lily have all of the same synapomorphies, and they have exactly the same synapomorphies. They both lack the reticulated veins. So we know then that grass and lily are sister taxa. So then finally we look over and we see that rose and sunflower, they have the exact same synapomorphies. They're both missing the parallel veins. So rose and sunflower are our sister taxa. Okay, did that make sense at all?
Yes, it made a lot of sense.
Okay, awesome. And then you should be able to answer the questions off of that. If you need help, let me know. Otherwise, they're kind of pretty straightforward questions, and he has answers to them listed. So.
Okay. Thank you so much.
Sure thing.
Thank you.
That's everything. Then I'm going to sign off for the night. Yes, I'm so glad you guys that it helped.
Have a good night.
You too. Good luck tomorrow.
Thank you.